let's continue our discussion on introduction to biotechnology. Today's discussion I will divide into two parts. The first one is emerging trend in medical biotechnology, and the second one is a product of medical biotechnology. Now let's start with the emerging trend. Stem cells are undifferentiated or partially differentiated cells that can differentiate into various types of cells and proliferate independently to produce more of the same stem cell. All stem cells can self-renew, make copies of themselves, and differentiate develop into more specialized cells. There are three main types of stem cell: embryonic stem cell, adult stem cell, and induced pluripotent stem cell. The first is embryonic stem cell. This type of stem cell are pluripotent stem cell derived from inner cell mass of blastocyte and early stage pre-implantation embryo. Commonly, human embryos are reach to blastocyte state 4 to 5 days post-fertilization, at which they consist of 550 to 150 cells. The embryonic stem cells said to be pluripotent, which means they can change into any cell in this body. The figure shows is presented the specific characteristic of embryonic cell. Let's see this one. This is the origin of the cell, then cell renewal, then this one pluripotence, this one. The second type of stem cell are adult stem cell. Undifferentiated cells are found throughout the body after development that multiply by cell division to replenish dying cell and regenerate damaged tissue. Adult stem cells are said to be multipotent, which means they can only change into some cell in body, not any cell. For example, blood stem cell only replace the various type of cell in the blood. Skin, this one is the epithelial stem cell, provide the different type of cell that make up our skin and cell. The most used adult stem cell are hematopoietic stem cell and mesenchymal stem cell. Let's see this one. Have a look to this figure. This is adult stem cell. This can be blood, muscle, brain, and others, especially the skin. Induced pluripotent stem cell are type of pluripotent stem cell derived from adult somatic cell. They have reprogrammed through inducing gene and factor to be pluripotent. Induced mean that they are made in lab by taking normal adult cell like skin or blood cell and reprogramming them to become stem cell. Just like embryonic stem cell, they are pluripotent, so they can develop into any cell type. This figure is present a schematic for human-induced pluripotent stem cell-based disease modeling. The creation and use of human-induced pluripotent stem cell, or you call that one as an EPSC, based disease model typically involve the following step. The first one, EPCG are derived from individual pattern, and isogenic control are created using gene editing technology such as CRISPR-Cas9. The induced pluripotent stem cell are then differentiated into specific stem cell such as neural cell, and the resulting cells are studied to identify disease-specific phenotype. Investigation of this phenotype at the molecular level can allow the identification of new pathological mechanisms, providing opportunity for drug discovery and personalized medicine. Now let's see this figure, a schematic for human EPSC. We start from this one, this point. Let's see this one. This is the pattern. Reprogramming a somatic cell. Then we'll see this one. Representing into desired cell type. Then we got the cell that we need by identifying that one with several tools. Then let's move to the Human Genome Project. The Human Genome Project, or we call that one as a HGP, was the international collaborative research program whose goal was the complete mapping and understanding of all the genes of human being. The primary work of the project is to develop the investigator to identify a gene involved in normal biology as well as in both rare and common disease. The finished sequence produced by Human Genome Project cover about 99% of the human genome gene-containing region, and it has been sequenced to an accuracy of 99.99%. In addition, to help researchers better understand the meaning of human genetic instruction book, the project took on a wide range of other goals, from sequencing the genome of model organism to developing a new technology to study whole genome. The Human Genome Project was an international research project that makes use of two methods that aim to identifying cDNA and the location of the coding as well as non-coding region of gene, followed by assigning a function to those genes. There are two methodologies that took part in the Human Genome Project, such as expression sequence tag and sequence annotation. Express sequence tag, they are short subsequent of cDNA sequence, and the methodology involving express sequence tag focus on identifying all the genes that are expressed as RNA. Sequence annotation, it is the process of identifying the location of gene and all the coding region in the genome and further determining the role of those genes. Thus, this methodology was used for sequencing the whole set of the genome and later assigning different regions in the sequence with a function. The Human Genome Project application, such as molecular medicine, was a control and environmental cleanup and DNA identification for a forensic field. Now let's move to recombinant DNA technology. Recombinant DNA technology comprise altering genetic material outside an organism to obtain, enhance, and desired characteristics in living organisms or as their product. This technology involves the insertion of DNA fragment from a variety of sources having a desired gene sequence via appropriate vector. The most common application of recombinant DNA is in basic research, in which the technology is important to most current work in the biological and biomedical science. Recombinant DNA is used to identify, map, and sequence genes and to determine their function. RDNA probes are employed in analyzing gene expression within individual cells and the tissue of whole organism. Recombinant proteins are widely used as reagent in laboratory experiment and to generate antibody probe for examining protein synthesis within cell and organism. The additional practical application of recombinant DNA are found in industry, food production, human and veterinary medicine, agriculture, and bioengineering. In general, recombinant technology have several steps, such as, let's see this figure, cutting the desired DNA by restriction site, amplifying the gene copy by PCR, inserting the gene into the vector, transferring the vector into the host organism, and obtaining the product of recombinant gene. This figure shows the schematic of how does the recombinant DNA technology work. Let's see. The principle of recombinant DNA technology involve 4 steps, as I have already mentioned before to you. I'll repeat it one more time. Cutting vector in this side, then insert the gene that you want to express to be something, vector containing a target gene. This is host cell, and this cell will amplify, then also will amplify the product that we want to have. Now, let's move to product of medical biotechnology. There are many products of medical biotechnology. There are some known products of medical biotechnology, such as antibiotic, vaccine, etc. Antibiotic are type of antimicrobial product used for the treatment and prevention of bacterial infection. Antibiotic can kill or inhibit bacterial growth, and the major mechanism underlying antibiotic is either interference with the structure of the cell wall of the bacteria or parasite, or prevention of them from multiplying. Antibiotics are mainly used for the treatment and prevention of infection. Antibiotics can be used to treat bacterial infection, protozoan infection, and immunomodulation. They can also be used for preventing infection after surgical wound and dental wound. Antibiotics are categorized based on their mechanism of function. Antibiotic function either against specific bacteria or against a parasite. A major type of antibiotic include penicillin, for example, penicillin, flucloxacillin, and amoxicillin. Cephalosporin, for example, cephaloxil, and cephalaxin. Tetracycline, aminoglycoside, clindamycin, sulfonamide, and trimipropin, and metronidazole and tinidazole, unilone, for example, levaprosacin and norprosacin. Now let's move to recombinant protein. Recombinant protein are a protein encoded by recombinant DNA that has been cloned an expression factor. That has been cloned in an expression factor that support expression of the gene and translation of messenger RNA. The first recombinant protein used in treatment was recombinant human insulin in 1982. Several recombinant proteins which had been produced and commercialized. Let's see this one. Let's see one by one. Humulin, this insulin, this for a diabetes. So many. You can see this one. Embryol on top for a cancer. Then now let's move to hybridoma technology. Hybridoma technology is a well-established method to produce monoclonal antibody, or we call that was as MABS, specific to antigen of interest. Hybridoma cell line are formed via fusion between a short-type antibody producing B cell and immortal myeloma cell. Each hybridoma constitutively express a large amount of one specific monoclonal antibody, and before hybridoma cell line can be cryopreserved for long-lasting monoclonal antibody production. As a result, researchers usually prepare generating hybridoma over other monoclonal antibody production method to maintain a convenient, never-ending supply of important monoclonal antibody. This figure, let's see in the right side, shows how the monoclonal antibody produced by hybridoma technology. Let's see this one. Immunization, antigen, then we got, after pass this step, we got this product. Hybridoma technology is composed of several technical procedures, including antigen preparation, animal immunization, cell fusion, hybridoma screening, and subcloning, as well as characterization of and production of specific antibody. Monoclonal antibody are widely used for the prevention, diagnosis, and treatment of disease. For example, the first one is antibody to prostate-specific antigen, placental alkaline phosphatase, human chorionic gonadotropin, and others. Monoclonal antibody for cytokinin 18 and prostate-specific antigen have also been developed for the diagnosis of prostate cancer. Monoclonal antibody have also been for the treatment of cancer and autoimmune disease. The major mechanism underlying this treatment include blocking of targeted molecular function, including apoptosis and modulating signaling pathway. During cancer treatment, monoclonal antibody can bind to cancer cell-specific antigen, activate apoptosis, and induce an immunological response. They can also use to deliver a toxin, radioisotope, cytokine, or other conjugate. In autoimmune disease, they are used to neutralize interleukin such as TNF-alpha, IL-2, or antibodies such as immunoglobulin A. Let's see once more time about this one. We have this antigen, stem cell, fusion, culture, then propagate, and the last of this process is harvest monoclonal antibody. Then now let's move to a vaccine. Vaccine is a biological product that aims to provide acquired immune for the particular disease. Commonly, a vaccine contains the entire disease-causing pathogen as a weaken or kill a pathogen or only their toxin or their surface protein. This agent will actively stimulate the host immune system that then produce a protective response to this agent. Several types of vaccine with its development method and some example of vaccine we can see in this table. This one, inactivated vaccine. It can be used for influenza, cholera, attenuated vaccine, toxin, subunit vaccine. Then let's move to the last one. This is also a stem cell therapy. Medical biotechnology has provided multiple technology and product for healthcare. With discovery of gene, protein, and stem cell, medical biotechnology has become an important bridge between biotechnology, medicine, and pharmacology. Molecular biotechnology, personalized medicine, and regenerative medicine have branched out of medical biotechnology to generate a new era of healthcare science. With this breakthrough, medical biotechnology is positioned to become the most significant field that improves human health and the quality of life. I think that's all for this moment. Thank you so much for your attention and take care. Thank you.